So we are celebrating the most cited and downloaded authors of 2016 and 2017 in the ASLO family journals. Okay, so our next invited author and speaker is Pat Glibert for the paper that you see in front of you, Pluses and Minuses of Ammonium and Nitrate Uptake, by Glibert and co-authors. That was published in LNO in 2016. There you go. So welcome, Pat.
Well, thank you for that introduction. Thank you, ASLO, for the invitation to be here and for putting on this special session. I want to talk to you today about our review paper on ammonium and nitrate uptake and assimilation by phytoplankton, a topic that we've known a lot about for a long time, but the question about how these relationships change with our emphasis on nitrogen-enriched conditions has come into increasing emphasis. So I do want to thank all of my co-authors who contributed substantially to this effort. So we know that we are enriching both freshwater, marine waters, coastal systems with lots of nutrients, but as we enrich with nutrients, we aren't necessarily enriching with the same type of nitrogen. So the gradient from chemically reduced to chemically oxidized nitrogen can change. For example, in Maryland coastal bays, we've seen a large increase in ammonium concentration over time. Same is true for San Francisco Bay Delta. I could put any of a number of examples here showing this trend in ammonium pollution, and we're seeing more atmospheric ammonium pollution as well, which ultimately winds up being aquatic ammonium pollution. So this raises the question, how does the uptake and assimilation of ammonium differ from that of nitrate? Are there differences between phytoplankton functional groups in the use of one form or another? And how does that change across concentration gradients? The key question that's being raised, of course, in eutrophic systems. We know that algae have a range of nitrogen sources they can use, not just ammonium and nitrate. There's urea, there's plenty of organic substrates that they can use. The cyanobacteria, some of the cyanobacteria, as we've just heard, can use nitrogen gas. But the use of any nitrogen form other than ammonium has to be converted to ammonium before it's assimilated into amino acids and proteins. So it takes more energy to do that. And so ammonium's long been considered the preferred nitrogen form. It's classically considered the preferred nitrogen form also because in classic laboratory experiments, if we put equimolar concentrations of ammonium and nitrate in the culture draws down ammonium first, and only after ammonium is drawn down does nitrate get utilized. So this has long led to the paradigm that cell growth on ammonium should be faster than that on nitrate because it takes more time to do those extra manipulation steps. But I'm going to show that this is perhaps not always the case. We also have another paradigm or dogma that has long been held in the literature, that is there should be no selective effect that might distinguish between the performance of any algae so long as nutrients are at saturating concentrations. In other words, nutrients are only regulating if they're in limiting concentrations. And again, I'm going to suggest that this isn't necessarily a consistent dogma across the entire concentration gradient. So we've long known, and I'm just going to go through a few examples of how we've known what we know about the regulation of ammonium and nitrate and how they differ. There are differences in temperature. If we increase temperature, typically ammonium uptake goes up, whereas if we increase temperature, typically nitrate uptake goes down, and this is due to the different temperature optima of their respective enzymes. We also know that if we increase ammonium concentration, the capacity for ammonium uptake often goes down. The number of transporters become reduced, and that is the cell protects itself from over-enrichment of ammonium, which in the cell can be toxic. On the other hand, for nitrate, as we increase the concentration, the capacity for nitrate uptake shifts up. So the shift-up phenomenon is one that is very well described in the literature. We've also long known that there are interactions between ammonium and nitrate assimilation. We add more and more ammonium, and the capacity for uptake, transport, and assimilation goes down. Ammonium interferes with transport as well as nitrate reductase. On the other hand, if we add nitrate, the capacity for an assimilation of ammonium changes very little. So the end result is that the kinetic relationships for these two substrates are really quite different. On the one hand, we have a preference for ammonium at low concentrations. It's taken up very readily and probably more rapidly than nitrate. But as we increase ammonium, we run into this repressive range and at very high concentrations, toxicity. So this kinetic relationship is not unlike that which has been well described for photosynthesis, photoinhibitory term here. For nitrate, we rarely see that inhibitory term. We often see a kinetic relationship that may accelerate or show a biphasic type of pattern. So how do these relationships change, or how are they different for different phytoplankton species? We know diatoms have a very large capacity to take up and assimilate nitrate. They've got lots of nitrate transporters on the cell surface. They've got a very large internal pool of nitrate. So why? So I'm going to tell you that nitrate is used in diatoms, as well as in some other types of phytoplankton, in a dissimilatory capacity in order for them to regulate cell energy balance. And this is particularly true in cool waters, and we know that diatoms are most common in upwelling areas, spring blooms, and it's in cool waters that nitrate reductase does very well. Nitrate reductase likes it cool. On the other hand, the carbon assimilation enzyme ribisco likes it warm. So we have a disconnect between the temperature optima of these two assimilating enzymes. And along the electron transport chain, this leads to a bottleneck. And this can do the actual damage in the cell when these two physiological pathways become uncoupled. And so cells have a number of mechanisms, overflow mechanisms, that allow them to have safety valves, as it were, to protect downstream metabolism. And in diatoms, this dissimilatory nitrate reduction to ammonium is one of these pathways. It occurs in the light reactions of photosynthesis here. This ammonium is produced in the chloroplast, but not all of it goes to nitrogen assimilation. Some get spun off from the cell, and we think of this as this overflow metabolism. There are many other overflow metabolic pathways. Maillard reaction here is one that's more commonly used by cyanobacteria. Other mechanisms such as photorespiration are also can be thought of as part of this overflow metabolism. So when we have lots of ammonium in the environment, we start to knock out this nitrate transport, nitrate assimilation, and therefore diatoms disproportionately can become repressed. So what about Pika cyanobacteria or dinoflagellates? Pika cyanobacteria generally don't have the problem of toxic ammonium buildup in the cell because they're so small, and so they don't have there's just not enough buildup of ammonium for toxic concentrations. Dinoflagellates are interesting in many ways. They have lots of ammonium transporters. They have lots of ways in which they assimilate ammonium in the cell. And lots of dinoflagellates we know are mixotrophic. They're taking in large particles, and they're digesting those particles. So they have many pathways to accommodate this ammonium. And so as we shift this gradient of chemically reduced to chemically oxidized nitrogen, we get a different community assemblage. More diatoms under nitrate, also larger cells under ammonium, more Pika cyanobacteria, some mixotrophic dinos, some cryptophytes, and in freshwater, more chlorophytes. So now I'd like to take the next few minutes to talk about a specific example where this has been shown in long-term data. So this is work by Vanessa Swarbrick, which has just been published in LNO, and it's work that was conducted in the Capel Lakes region of Canada. Here I'm focusing on Wascana Lake, and 16 years of bioassays were done with ammonium enrichment. All done over the 16 years exactly the same, and they were done to test the extent to which these lakes were nitrogen limited. But over the 16 years, this lake has changed. Shown here in blue lines are the 1990s data, and as it gets more orange, the more recent data, and this is just chlorophyll, and you can see the eutrophication of the lake, much more chlorophyll now in recent years. And this chlorophyll is composed in the spring, more diatoms, and in the summer, more non-nitrogen fixing cyanobacteria based on pigment composition. So what about the ammonium enrichment bioassays? Shown here is the percent change in the chlorophyll relative to control, the amended chlorophyll relative to control. In the 1990s, adding ammonium in the spring led to a 20 to 30% increase in productivity. So these were nitrogen-limited systems. In the summer, in the 1990s, very little change. More recently now, adding ammonium leads to a 20 to 30% decline in chlorophyll, that repressive effect, and in the summer, a large stimulation. So a big shift in the effect of these ammonium amendments. So basically what's happening is adding ammonium now is knocking out these diatoms, and in the summer enhancing these picocyanobacteria, or non-nitrogen fixing cyanobacteria. So just to conclude, I think I've shown you that ammonium can be repressive at high concentrations while being a preferred nitrogen substrate under low concentrations, and this repressive effect is becoming more and more important in our eutrophic environments. Ammonium and nitrate are assimilated differently by different phytoplankton groups. We've long known this, and the sensitivity to ammonium repression affects phytoplankton taxa differently. Now, what are a couple of implications of this? One is that in developing nutrient criteria for our fresh and coastal waters, we rarely take into account nitrogen form. We're still developing nutrient criteria that quantify N as total N, and we need to work to incorporate these different forms into nutrient criteria. This is going to be an ongoing challenge. But we also know that the development of many mechanistic models, both global and regional models, are now incorporating ammonium inhibitory terms, and the incorporation of these ammonium inhibitory terms in both the global and regional models is leading to better correspondence of data with historical patterns as well as better predictions going forward. So a plug to continue to incorporate these ammonium inhibitory terms in models as we go forward. So just to conclude, I want to thank ASLO again for this invitation. I want to thank all of my co-authors for their contributions to the work I've talked about today. Thank you.